Okay, let's get a few slides in Chapter 9. So the topic in Chapter 9 is it's looking at chemical bonding. And the big thing that to get out of Chapter 9, I'll tell you, it's really important to take the information out of Chapter 9 because it's the foundation of Lewis theory of what you're going to do in organic. So all of you that are planning to go through organic chemistry as a support course or as part of the double major, this is a foundations chapter that you just need to get. If you're going to spend the time over the Thanksgiving break and read this chapter. Okay. There's a lot of things introduced here. We're going to talk about the types of bonds, introduce the Lewis dot notation, talk about ionic bonding for a little bit and what happens when we have this electron transfer. Then we're going to look at covalent bonding and talking about electronegativity, bond polarity, because bonds are not always sharing electrons equally. That's critically important. That's what drives protein structure, the unequal sharing of electrons. Okay. Then we're going to go into the Lewis structure of molecules. This is just this is the rest of chemistry. Is the foundation of understanding Lewis structures. It talks about when you have more than one possibility. That's what resonance structure is about. Formal charges are a way that we figure out which is the most probable structure when you have multiple options. And then we're going to talk about some of the exceptions to the octet rule, things that want 8 electrons we talked about in the last chapter, and not everything can get there. Okay, so some things are stable without 8 electrons and exist in nature. And then the last thing they're looking at bond lengths and bond energies in metals. Okay, so why does things like this matter? So as I told you, if you sit here, you're going to get loaded up on good questions. Okay, give me an answer. 3, 2.
Just a no to true.
Yeah.
Tired of this nonsense.
There you go. Dot dot click.
I don't know why.
You found a way around it.
The channel off.
Chose C.
There's 16.
Yeah. So 2 people hadn't done it.
You're the only one who's going to get it wrong.
There's one more. Okay. Woohoo. All right. So if we look at where this becomes important, things that are problems in the world today, certainly 30 years ago, HIV was considered an epidemic that was a threat to influence world population. 15% of the continent of Africa died to HIV virus. It spread to the United States in the late 1970s, early 1980s. It was fairly contained and then started to spread in the United States significantly. Hundreds of thousands of people have died to AIDS in the United States alone. More than likely, you know somebody. Okay? I do. I know several people that died of it. So one of the ways that we've tried to stop this is look at the HIV protease. The HIV protease is a protein that's synthesized by the virus itself. And this particular protein is crucial because it controls the ability of the virus to multiply, and which leads to the autoimmunodeficiency syndrome, which is known as AIDS. And AIDS is what actually causes the death. Basically, it shuts down the body's complete self-defense systems. Okay, you die of common infections. You get pneumonia and can't ever recover from it, or whatever it may be. So, during the start of this, pharmaceutical companies are looking at it as it's a double-edged sword. Pharmaceutical companies are medical entities, and they're looking at they're under a type of oath like a doctor's when they become a pharmaceutical company, is that they're working in the pursuit of saving lives. On the other side of it, most pharmaceutical companies, especially in the 1980s and early 1990s, there were startup pharmaceutical companies that were started with the creation of making bucks. Okay, so they had visions of making money, and people would put in a lot of money to startups. They would put in $100,000, and they'd get from 1,000 investors, and all of a sudden you got $100 million, and you could start a pharmaceutical company, hoping you get the magic drug, the magic pill that changes human health, and then you could make billions of dollars selling that drug. One of the companies that I worked with, I worked for, when I got out of graduate school, I worked for a company called Monsanto in St. Louis. We had a pharmaceutical company that was named Serum. And one of the projects we worked on was the development of an HIV protease inhibitor that would shut down this protease. And if we could figure out how to control the protease, we could have something that could be a potential cure for AIDS. Every pharmaceutical company was working on this common cause. They tried the way that we did this is they worked out the crystal structure of the protease, and they knew its exact three-dimensional shape, and they devised these compounds called inhibitors that would bind to this active site, and we use the terminology in upper level courses, a lock and key mechanism. So you've got something on the surface that has a shape associated with it. If you can design a molecule with an exact shape that would bind to this tightly, so if its shape's right, it could bind to it so tightly, it could block this from the next compound that's supposed to go in there in the natural process. This would be blocked. That's the way inhibitors work. And that's a very simplistic mechanism, our explanation of it, but that's the summation of what these HIV inhibitors were trying to do. They were trying to block this protease from working, that molecular machine. So there was a ton of research that was done into finding out what is the three-dimensional structure of all the compounds of the proteins involved with AIDS, and how can we block this. There were tons of potential molecules that were developed with different shapes, some done with trial and error, some done with computer-aided simulation. You mentioned Frederick, Maryland. In the Frederick, Maryland, I went to the National Cancer Institute to a conference and presented data that we were doing. And one of the things that they talked about is there's a supercomputer at Fort Detrick. So Fort Detrick is a kind of a place with a split history. So things that you'll hear about Fort Detrick is home of the nation's chemical and biological warfare agent. All of our chemical and biological warfare agents were developed there. Now the protection against those are developed there. The anthrax that was smuggled out and sent to the senators and killed people in the same time as 9/11, that same time period, that anthrax was smuggled out of Fort Detrick. Okay, it's where the worst of the worst in terms of biological species are in the United States. It's also home to the National Cancer Institute. Same facilities. Okay, kind of crazy to me, but there's a supercomputer there, and it's one of the largest supercomputers at the time. That supercomputer is probably the size of this floor, the entire floor of this building, and the whole building is immersed in liquid nitrogen to keep the supercomputer cold. And it does at that time was doing some of the most powerful simulations that could be done. The HIV protease protein was one of the largest molecules that initially did a protein modeling on to try to figure out what do we think its structure actually looks like? What's our best idea? And how can we go after it to try to inhibit it, stop it from doing what it's doing? So that's what that conference was talking about. One of the companies that drugs that our company developed at that time was had very strong inhibition of the HIV protease, so it would go in here and it would bind it very strongly, and in a petri dish it would stop it from working. Okay, it would shut down the HIV protease. The problem was when you put it inside of a test animal, so when you put it inside of a living cell system, its efficiency went way down. And the project I actually did on it was trying to figure out, is it binding to something else in the plasma? And I tested against all the different common proteins and found that it was binding to acid-1 glycoproteins, which are one of the top 3 proteins in plasma. And that was, the drug wasn't getting to the protease, it was sticking to something else where it got to do what it needed to do. So at that time, the company was starting to split some things off, but that product that we were working on was sold to Merck. Merck bought Cyril out, and today that's part of the cocktail. So that the parent to that drug that's used today, Merck made one modification on it on the structure, and this is part of the way that we changed it or modified it. And today, AIDS is such a complex thing to try to stop. There's no way that, and there's the different ways. They're looking at it with nucleoside reverse transcriptase inhibitors, the non-nuclease reverse transcriptase inhibitors, the protease inhibitors. There's fusion inhibitors. Unless you've had upper level biology classes, these may not make a lot of sense. And integrase inhibitors, but there's 5 different approaches they're using, 5 different sets of drugs that are mixed together. Each one of them is trying to do a different thing to shut this down. What we find is that it mutates in the body so quickly, you can't always just go with one approach. But this cocktail or blend today is the best thing that we have to stop AIDS. And they're actually doing it now for the first time, giving this as this mixture as something to prevent AIDS, where you have children in very, very high risk situations, or they're giving it to children early on that may be infected with it before the virus gets a chance to, or AIDS gets a chance to fully develop.
What do you mean at risk for like mothers with HIV?
Yes. So if you're in populations where where AIDS represents 80% of the population, yes. So, and it's very controversial. Part of the reason is it's literally costing. Initially, a dose of this stuff cost $1,000, and a dose of it was going to give you protection for a couple of weeks, and that was it. So you're trying to protect a population. You were spending billions and billions of dollars. So one of the arguments is, well, let's let those that have AIDS die off and kill it out, just keep other people from getting infected. It doesn't work like that. Okay, and especially in the case where AIDS was spreading most rapidly in Africa, there were a lot of cases where it wasn't consensual sex that was causing this. And there's a lot of rape, abuse, different things, slavery, things that are going on that still was going to cause this disease to transmit. But today, this is the best that science has been able to come up with in over 30 years of study. There may be a magic pill that annihilates AIDS in the future, and it disappears. We're using the same thing to stop other things that come in. So, has there been anything in the last 3 years that you can think of similar to AIDS that's been large-scale health threat in the United States?
Diabetes.
Okay, diabetes is certainly, and we're the Oklahoma's the heart of it. I'm a type 2 diabetic, so I stick my finger every day. But I'm talking about something in terms of and diabetes is an epidemic, without a doubt. It affects a larger population or percentage of the population than any other disease in the United States right now. Heart disease overtook heart disease. Does anybody remember a situation where a person was affected with Ebola, and then they came into Fort Worth, came into the Dallas Metroplex, and that person got the nurse that was working on her infected there too? We spent a huge, huge effort to stop that epidemic from becoming widespread, because we don't have the tool set to deal with Ebola like we do with AIDS at this point. I mean, and it rapidly changes. But the future of fighting off those infections that are a threat to humankind are going to be in modeling, okay? And that stuff is going to take time. The thing that's changed so much, when I was in graduate school in the early 1990s, late 1880s, if a person worked out a protein sequence to discover a new protein, just figure out what that was 5 years of work. And if another person isolated that protein and got the crystal X-ray structure of it, that was another doctoral. It's a 5-year PhD study. And today, technology's changed so much that they can get a DNA sequence on a new organism and get a crystal structure in the same day, and try to figure out, model this thing to look at a target. There's so many different things out there that we're looking at, and we've got rapid ability to approach it. We use a term called data mining. We're producing so much information, and there's so much things out there, is trying to figure out how to pair things up, figure out what's important and what's not. There's got to be a fusing of what's happening in the technology, the things that we're measuring responses and looking at things, and the ability to sort through that information. That's going to have to come through computer sciences. There's just more things that can be done. I worked on a modeling program with a group from Virginia Tech and colleagues at the Noble Foundation, where we were looking at a model plant system. It was called Medicago trincatula, and it's looking a lot like alfalfa, for those of you who know what alfalfa is. And they were taking these cells, and they were looking at these cells, how they change over time, and they split the cells up, and they gave a bunch of different groups. So my group looked at amino acid concentrations of sugars and ions that are present. And they did these as a function of time, as a control, and then they did it with exposure to UV light, exposure to drought, exposure to funguses and different types of things. And like my group was analyzing each cell, they were coming up with 250 data points. So they were measuring what's the concentration of all these sugars, what's the concentration of all the amino acids, all these different ions. The protein group was coming up with 4,000 data points. At the end of this, we had hundreds of thousands of different data points that were tracking as a function of time, and you're trying to look at the correlation of what's related to what. The idea behind this is they were trying to develop a computer model that completely represented what's going on inside the cell. Okay, they were trying to simulate what's going on in a cell. And we've been trying to do that literally for 50 years. And this study was overall failure because, it developed new steps, new strategies, but there ended up being more data than we could process. And there were some neat discoveries that were made from it, but in terms of the global modeling, which was the overall proposal, Virginia Tech wasn't successful to produce a model that represented a cellular system. So, okay, 16 quickly. Or not. Okay, I'm going with somebody left then. That's only 15. Okay. All right, so there's a lot of things that we need to understand on how things are attached together, how atoms are bonded to one another. So one of the things the chapter starts with is trying to figure out the how and why. How are two atoms connected together, and why do they connect? It wants to explain why some combinations of atoms are stable and others are not. Why is the most abundant molecular compound on the surface Earth water, and why not HO or H3O? Those two elements were obviously present in high abundance. So why did they come together in those combinations? The key thing is we want to learn from this how to predict shape, because shape turns into function. If we understand the shape of molecules, we can understand what's going on with the function of. From those, we can learn how to predict the physical and chemical properties. So the person that this chapter is focused around was G.N. Lewis, and he came up with a model that is a very simple approximation of explaining bonding theories. There are much more complicated and complex bonding models that work. This is the foundations. So if you take more advanced chemistry classes, you'll learn about other bonding models that explain things in more detail and can give better approximations, but you have to start somewhere. And this is what we think is a good starting point, and all the textbooks use this as the basis for how we start to describe things. It focuses on those outer shell electrons we talked about in the last chapter, valence electrons. Those are the important ones, the outer layer of the onion. Okay, it doesn't really matter what's underneath, it's that outer layer that's chemical reactivity. And from this, we can draw models, that we call Lewis structures, and we're going to develop what's called electron dot structures. There was a question about that a week or so ago. And from this, we can start to predict properties like stability, shape, size, and polarity. And those are the critical things that influence whether something's going to be a good inhibitor, where it's going to be a good drug, what its function is going to be. So the first question is why do atoms bond? And the reason is they get a lower potential energy between the bonded particles than the atoms by itself. And the way I tell you to think about it, let's say that you had a marble and you drop that marble into a bowl. So you drop it in the bowl, and what's it going to do? It's going to start going back and forth, and you can amuse yourself for however long you want to sit and watch it. But at the end, you know where you're going to pick the marble up. It's going to be in the bottom of the bowl. Okay? And the reason it's going to be in the bottom of the bowl is because it's going to the area of the lowest potential energy. So the bottom of the bowl represents the lowest potential energy associated with that. And here it's gravitational potential energy. But that same concept of why the marble going to the bottom of the bowl or a brick falling to the ground or whatever you want to think of happening is driven by energy is because it's going to a lower potential energy level. It's the same thing that's happening with atoms when atoms combine. They're combining because there's a lower potential energy at the end. Okay, and they're always looking to form the lowest potential energy that's available in the system. All right, so if we look about this and think about the things that are involved to calculate potential energy, things that we have to consider are the repulsions that happen from the nucleus. Those are like charges. The repulsions that happen from neighboring electrons, they're like charges. And then finally, the attractions between the positively charged nucleus and the negatively charged electron. So it's a complex competition. So from this, there are three types of bonds, and we've already talked about these types of bonds that we can classify the connection between atoms. And those are ionic, and those happens between metals and nonmetals. Covalent, between nonmetals and nonmetals. And this is where electrons are shared. Ionics are where electrons are transferred. And metallic are the bonds that exist between metal atoms. And the term that we use is pooled. The electrons are like a sea. Nobody really owns the electrons. They're just kind of floating around. And that's what gives metals the property of high electrical conductivity. Is the electrons are not really attached to any individual atom. They're just everywhere, floating around. Okay, so if we think about these types of bondings, again, we've talked about this a little bit already in the semester. Ionic bonds are the combinations of charged particles. Opposites attract. They come together in a crystal structure. Covalent bonding, like a water molecule. So you have the discrete unit of a hydrogen and two hydrogen and oxygen connected, and then they can come together to form an entity called the molecule. And then your last possibility, and the one we talk about the least in this course, is what happens in metals. And again, this maybe picture kind of illustrates it a little bit, is that sea of electrons that are there. Now, I talked about previously before the last exam, almost every chapter has a set of little introduction videos. The author, especially in chapter 9, he's got, so there's like 5 or 6 of those little videos in there. And they're a couple of minutes apiece. He does a good job of explaining that material to tell you what's going on. Okay, so, all right, so load up on them. Okay, all right, so who was gone a while ago? Did we step out? Something. Okay, so we've got those. Okay, so I think we're just going to call it there. So ionic bonds is where we're going to start. Start talking about those. Again, I encourage you, if you haven't been reading the book, read chapter 9. Okay? Say again?
So I know that you do like volunteer hours.
Yeah.
But it's like volunteer.
they look for people that are doing volunteering of a lot of different types. So if you want to, yes, balance that out with something else, anything that's you care for humanity in general, care for nature in general, conservation clubs, any of that stuff is a lot.
Okay.
So, Shane, I'll just tell you, it can't be only that. It's got to be most of those stuff.
Okay.
Yep.